what we're going to do is we're going to tell you about Entos, which really is a company built around AI-driven chemistry and the use of that AI-driven chemistry platform for the development of better small molecule therapeutics. The core capability of Entos and the core goal of Entos is really to make better molecules faster. As you'll hear today, we have a differentiated and extremely powerful machine learning AI platform that allows us to make better predictions across many different endpoints that are essential for drug discovery with very sparse experimental data. And that allows us to very efficiently explore chemical space. Chemical space is extraordinarily vast. You're always on the search for better molecules that will be drug candidates. And being able to push through that space and to reliably guide the search through machine learning technologies in a multidimensional way is an extremely powerful aspect of what we bring in terms of our design capabilities. We combine that with high throughput automated chemistries so that those predictions can be rapidly converted through parallel chemistry and through high throughput flexible chemistries into physical reality. And we have novel technologies for how we can take those reaction mixtures that are executed in parallel and to rapidly assay those both for biochemical and cellular readouts. And that allows for an exceedingly fast design, make, test cycle where we can discover and rapidly optimize new molecules for indications with substantial unmet patient need. And we're really doing that in a very focused way, as you'll see, for the pursuit of solutions to pressing a therapeutic need in the space of oncology.
The core platform in Entos combines both an innovative and novel AI technology with high throughput experimental capabilities. I'm just going to walk you through those components and just give you a little look under the hood about how they work. The process of discovering drug molecules can be viewed as a multi-parameter optimization problem. It's an optimization problem that takes place in a large number of dimensions and with very complex constraints on the chemical matter that can be tested and explored. The key technology behind our AI platform is called ARPANET, and ARPANET really is an embodiment of the idea that by building in more physics into AI and machine learning models, one can make accurate predictions with access to smaller amounts of data. And that is a really critical theme and one that you'll see carry through the subsequent slides that gives us a differentiation against competitors using AI in this space. Now, in conventional machine learning technologies, you'd be describing those molecules in terms of connections between different kinds of atoms. And that means that, for example, you would be in a position where knowing everything about ammonia and hydrogen fluoride will tell you nothing about water. But you can see with your own eyes that if you use a particular class of features that are proprietary features used within Entos, features derived from the molecular orbitals, the quantum mechanical molecular orbitals of the molecule, that there's a very linear and continuous relationship between those features, irrespective of which molecule the feature comes from. And that singular fact allows us to very, very efficiently and compactly describe chemical space and make machine learning models that can learn from a very small number of data points.
No, I'm always just excited for humanity when hearing about the progress you guys are making. It's just super impressive.
Thanks.
One question that I think might be helpful for the audience to better understand is just I'm always curious about the origin story of a company. And so could you just perhaps provide some context as to how you met, why this is the company that you both decided to leave, academic careers for, which is a big deal, obviously, when you have your lab set up and everything else. Just provide some context, if you will, to the audience for just the origin story of Entos.
Absolutely. Happy to tell that story. I think it was probably East Lansing, Michigan, or somewhere like that we met in at some conference about a decade ago. We hit it off, Tom and we started talking science, and more or less immediately after, sort of going back to our respective places, Tom to Caltech, me to Bristol in the UK, we started collaborating on a scientific program. And that, you know, we collaborated for a long time. We wrote a lot of papers together and did a lot of science. But the real seed of the Entos story, I think, came when I was in Caltech for a sabbatical year. So that was 2013-14, if I recall. And the you know, the conversation, probably over beer, strayed to the question of how we could really work together to direct our sort of scientific capacities to the most important and tangible problems. And so that, I think that, you know, we didn't know we were going to start a company then. We didn't know it was going to be a drug discovery company. We did not know we were going to leave our academic careers. But that, for me at least, was when the first sort of inkling of a thought started. And then, you know, there's a long story that about how that got embellished into the company that you see today, and the decision that Tom and I took to fully sort of commit to that story, leaving our academic positions. But anyway, that for me at least is the early phase of the origin story.
So maybe you and Fred could just talk in simple terms about how I think about this, how you think about it in terms of moving things that had to be done at the bench, had to be done in vitro, moving them into what's often referred to now as in silico, so that you're able to speed up the process and produce, you know, higher quality candidates and output before you go back to the bench or go in vitro or in vivo.
Absolutely. I mean, that is genuinely the case, Kevin, that the technologies really enable us to miss out enormous amounts of bench work and miss out the synthesis of a whole lot of compounds that will never have a chance, you know, that are not kind of moving with the spear tip as you advance through chemical space. And that's partly achieved through the capabilities of the AI platform, but the, you know, some of these 1000Xs are very much in the wetware. So you know, it's a classic thing in software that you make things faster by parallelization. Well, that's exactly what we're doing on the chemistry and high throughput experimentation side. So instead of doing one reaction to make one molecule, you do 1,000 to make 1,000. And that parallelization is possible using robotic instrumentation and the coordination of all of that through software. And really, you know, I mean, an important part of the differentiation of the platform is the ability to execute on that and really carry it all the way through from synthesis to these high value assay data points. So that's the sort of wetware end of the sort of reason that we can execute so effectively in drug discovery. And you know, we very much view it as, you know, although our background was more on the software and an AI and quantum side, we very much view it as the integration of two equal parts that mutually reinforce one another and bring out the maximum possible value from the complement. Yeah, so that's the way I think about the manner in which we're accelerating that discovery is through the integration of those two components.
There was a question I wanted to ask from the audience actually about the experimental data, or just maybe I'll just summarize this to say like operational question. You know, so when you first started this, you know, kind of maybe post A, right? You're starting to, you know, how much experimental data does it take to get going? And then how much work is it to structure and classify the neural network or the data to train on? You know, like what pre-work is required to organize the data to get going, essentially?
I'm very happy to talk about that because it turns out that that is a big deal. So you know, one of the reasons that it isn't easy for competitors to just you know, start doing this kind of stuff is precisely that issue of organizing data. I mean, there are lots of other differentiations, but that mere fact of organizing data is a huge task. So we were super lucky. We came into this, we already had a 25-person software and machine learning outfit when we made the decision that we were going to start doing this. And so, you know, we had this amazing opportunity to build a software architecture and a data architecture in advance of doing a ton of in-house experimentation. And that I think has converted to our ability to make this incredibly early start in AI-powered drug discovery. It can't be underestimated how much of a big deal processing and handling that data effectively actually is. So yeah, we're in a very strong position in that regard. And whenever new experiments are considered or designed, then always part of that consideration is how the data is going to end up in a database. We have frameworks, for example, that automatically register the existence of new data and automatically retrain the models. And so that's all dashboard, it's all available to the medicinal chemists and to others in the company. So we have a huge amount of automated technology that enables us to move really briskly as new experimental data get read out.
Yeah. And then there's another question that I think hits on another operational question, which is, are there bright red lines for Entos in terms of things that you'll do or won't do as a company? So just, you know, the clinical trials, internal manufacturing, you know, or are you trying to, you're taking it one step at a time and you know, either way, I'm just curious if you've drawn bright red lines for the company as you kind of now reach this next phase.
Yeah, I can take a crack at that. You know, you never say never, and you don't draw a lot of bright red lines. However, you play to your differentiation, right? So we have demonstrated differentiation in certain parts of the drug development pipeline, and you know, that is the part of the pipeline where we can do it better than everybody else, and therefore we should really be doing it there. As those assets move out of that sweet spot, then, you know, at some point you're doing the same thing, the best practices that everybody else is doing. So then you no longer have that core differentiation, and that, you know, begins to be a more obvious case for when you would then, you know, engage in partnerships and in collaborations when you're not differentiating the way you're doing it yourself. Having said that, you know, the Entos platform is not a static thing. We weren't differentiated for in vivo studies a year ago, and you know, and now we really are. So, you know, the platform continues to grow and to innovate at the pace of the development of the assets themselves. So, you know, and we're excited to see how far we can continue to push that and to continue innovating, and have every intention of doing that. But you're right. At some point, you know, we're going to be, you know, putting them on boxes and marketing and all that stuff. And I suspect by that point we'll definitely have partnered.